Hello, thank you for tuning in to this Young Women in Bio online event. Today's presentation is organized by Young Women in YWIB-RTP. We are a volunteer group from the Research Triangle area of North Carolina that plans STEM-related activities for girls in grades K through 12 throughout the year. We hope that by attending today's presentation, you will learn about what happens in clinical trials and why technology is needed to help speed up the process. This virtual webinar is part of our brand new Young Women in Bio online platform, which provides young women across the United States and in Canada free access to our educational and leadership programs, as well as other opportunities in science, technology, engineering, and math. Young Women in Bio is launching this new initiative for young women who are curious about STEM and want to learn more. This platform is particularly useful for girls who may not live near one of our 13 Women in Bio chapters in cities across the United States and in Montreal, Canada. Young Women in Bio, also known as YWIB, gives girls today the inspiration and support they need to become tomorrow's leaders in science, technology, engineering, and math. We strive to provide educational and hands-on experiences in STEM and hope to share our passion for all scientific fields. The launch of the Young Women in Bio online platform comes at a time when there is a real need to provide girls who can't otherwise attend our events with access to online programming in STEM. This will help with our goal to ignite curiosity and to inspire young women to see a future for themselves in STEM. We believe that by making these online resources available to a broader audience of girls, we can encourage more young women to find a lifelong passion and career in STEM. Young Women in Bio is part of the Women in Bio organization that has chapters nationwide and in Canada. Women in Bio is a nonprofit volunteer organization that promotes diversity and inclusion for women in the life sciences and beyond. There are currently 13 Young Women in Bio chapters across the United States and in Canada. To learn more, please visit the website listed on the screen. Today's webinar is sponsored by Datacubed Health. Datacubed Health is a pioneering technology company whose goal is to make better science and healthier communities a reality. Datacubed Health hopes to apply individualized solutions to collect data from patients in virtual clinical studies using remote engagement. Through today's webinar, we hope you will learn how clinical trials are performed. You will also learn how technology, like mobile apps, is revolutionizing remote data capture for clinical trials. Today's discussion will be led by two speakers from Datacubed Health. Our first speaker is Jennifer Cho, who is the Deputy Chief Scientific Officer at Datacubed Health. Jennifer studied child development and community health at Tufts University. She received her Doctor of Pharmacy degree from the University of Maryland. Jennifer has lots of experience working in clinical trials. Most of her work has focused mainly on cancer therapeutics. Jennifer has worked both in a hospital and a pharmaceutical company setting. Our second speaker is Pooja Rajguru, who is the lead project manager at Datacubed Health. Pooja studied biomedical engineering at Arizona State University. As a project manager, Pooja is responsible for the planning and execution of technological projects. She ensures project goals are met in a timely manner and that the project is meeting quality benchmarks. If you have questions at any time throughout the webinar, please feel free to ask your question in the chat box. To access the chat box, click on the question mark button in your dashboard within the GoToWebinar screen. At this time, I will pass it off to Jennifer, who will begin our discussion on how clinical trials are performed.
Can y'all see okay, I hope? Okay, so thank you so much to everyone for having us today and for joining for this webinar. So I did want to just start off by saying that, today we'll go through a little bit about drug development, drug approvals, and then talk a little bit about technology within the realm of drug development. And I did just want to start off by saying that drug development is a really, really big industry. And this is because there will unfortunately always be people who get sick and need medications. So there will always be lots of different and exciting career options within drug development that will help you to make a career out of helping patients. And sorry, before I move forward, I realize I'm going to just, sorry, I was going to try to make this full screen, but it seems to not be working very well. I hope you guys can see the slides okay. So just to give you a sense of how big of an industry drug development really is, wanted to give some context and mention that in the United States today, there are more than 20,000 approved prescription drugs and more than 100,000 over-the-counter drugs. So prescription drugs are ones that need to be prescribed by a doctor and dispensed by a pharmacist. And then over-the-counter drugs are anything that you or I could walk into a CVS and buy right off the shelf. So today's presentation is going to focus on prescription drugs and understanding essentially where these drugs come from and ultimately how do we get new drugs approved and on the market. So this slide is a general overview of, again, how specifically prescription drugs get approved. So pretty much every approved prescription drug out there follows this process. And the reason for this is that we want to make sure that all drugs are evaluated in the same way and that we're making informed decisions at each step of this process to make sure that we won't accidentally harm any people who might be taking this new drug. So I'll go into each of these steps in more detail over the next few slides. But for the most part, the overall idea is that first, within drug discovery, scientists are trying to brainstorm different molecular compounds that might be useful in treating a disease. Some of these compounds that they create will then get picked to be tested in a laboratory, and so this is called preclinical research. So that literally means before human research. And then so in the US specifically, the FDA would then review all of the preclinical data and give the okay to start testing this new drug in humans. And any studies that involve human testing are called clinical trials. So there's different phases of clinical trials, and I'll get into all of these in more detail, but essentially after phase 1, phase 2, phase 3 studies are conducted, then the FDA would review all of this data from human testing and then decide whether or not a drug would get approved. And so just to give a little bit more context around this, so within the drug discovery process, let's say scientists come up with 10,000 potential compounds that they think might work to treat a disease. On average, only one of these will actually end up getting approved by the FDA, and that whole process can take anywhere between 10 and 15 years. And so I've run, I used to work on clinical trials, and each of those trials can take years at a time. So the point here being to keep in mind as we move forward through this presentation is that drug development does take a very long time, and it's very expensive. Each study can be upwards of like many millions of dollars. So to go through the drug development process in a little bit more detail, like I mentioned, so the first step is drug discovery. So essentially at a molecular level, scientists understand that everything in the body happens through very specific cellular or protein pathways. And so based on what scientists know about a particular pathway, they can pick a part of that pathway and try to create new drugs to target that part of the pathway. So here on this slide, on the bottom left corner, this is a pathway that we know as a clotting pathway. So, you get a paper cut, your finger starts bleeding, and then after a minute, it stops bleeding because there's a clot there. So this is exactly what's happening in that minute. And so scientists know that there are different proteins involved. So what they've done is they've created a whole class of drugs to target each of these proteins. So for example, here, thrombin is a protein that's involved in continuing this clotting pathway. So scientists developed a whole class of drugs called direct thrombin inhibitors, which, they developed these drugs knowing how thrombin is shaped, and they created drugs that would fit into certain places on the thrombin protein to prevent it from working. So this is just an example of how, at a very molecular level, this is what informs drug discovery. So again, after drug discovery, where there's potentially, let's say, 10,000 compounds, a few hundred of those might end up going into preclinical research. So the first piece of preclinical research is in vitro research, and this literally means in Latin, within the glass. So the main goal of preclinical research is to test how specific cells would react to a drug. So this takes place within literally a petri dish. They put in drug, they put in cells, and they see what happens to the cells. And so this is the first part of preclinical research because since we really don't know what the drug is going to do, it would be unethical to just start trying it in actual living creatures. But based on the in vitro data, if that looks okay, then the next step for preclinical research would be in vivo research, and in Latin, this means in a living thing. So the goal of in vivo research is to understand how the drug is working in an actual living body. And so this is typically done in animals. Mice are most commonly used in the lab. They're pretty easy to, grow and manage. And obviously mice are not the same as humans, but there's enough similarities where scientists can make educated guesses about how the drug might work in humans. So again, after preclinical research, like I mentioned, in the US, the FDA would review all of the preclinical data and then give the okay to start clinical trials, so testing in humans. So the first step within this whole process is to do what's called a phase 1 clinical trial. So these are also known as first in man or first in human studies because this is literally the first time that this drug is being tested in humans. And so because it's the first time, these studies are very small, they don't involve a lot of people. So maybe we're talking about 10, 20 people on average. And a lot of times they're done on healthy individuals. And the goal is to really understand what the body does to the drug. So, how does the body absorb the drug, break it down, get rid of it? This is known as pharmacokinetics. And then what does the drug do to the body? So this is known as pharmacodynamics. And we can measure that by taking lots of different blood or urine samples and doing other clinical tests to the study volunteers. And the main goal of phase 1 trials is by far the biggest thing to understand is safety. Because again, this is the first time the drug is being tested in humans. So the other thing that we want to know within a phase 1 trial is what dose is appropriate in humans. So phase 1 studies usually test a variety of doses, and we're trying to find the right balance between for a dose that's not too toxic, but isn't too low that it's not going to have any effect in humans. So after phase 1 trials, if the drug looks safe enough, then we would move on to phase 2 trials. So in a phase 2 trial, it's a slightly larger group of people that would be involved, and these would actually be in patients who have the condition that this drug is trying to treat. And so the goal in phase 2 is still to evaluate safety, but here we're also starting to collect more data on how well does this drug work for the condition that it's being developed for. So once the phase 2 study is complete, then that drug would move on to being tested in a phase 3 trial. So phase 3 involves a lot more people, and how many depends really on the condition. So for example, I used to work on cancer studies, so some of these cancers are very rare, and so a phase 3 study might have 300 or 400 people, but a drug for blood pressure, like a lot of people out there have high blood pressure, so those studies might have like anywhere between 10 and 15,000 patients. But the idea is that you want to test in a bigger population to make sure the things that you were seeing in phase 1 and phase 2 weren't just a fluke, that with a bigger sample size, the drug is still behaving the way that you're expecting it to. And really, phase 3 is also, these studies are known as confirmatory studies because you want to confirm that the drug is actually treating the condition that it's being developed for. So phase 3 studies will often compare against medications that are currently being used to treat that particular condition and see whether this new drug is better than the currently available drugs. So again, after phase 1 and 2 is done, phase 1, 2, and 3 is done, the FDA would review all of this data, and then if they decide that it's effective and this drug is safe, then it would earn approval by the FDA. And then what we have after approval is what's known as a phase 4 study. So this is more of a real-world study where the drug is now out on the market, doctors are prescribing it, but data will still be collected to see, primarily to make sure that there aren't any new safety issues that are now coming up that were missed somehow in phase 1, 2, or 3 studies. And there have been a few instances of some drugs that got their FDA approval taken away from phase 4 data because there were new, very serious safety signals that were identified after the drug got approved. So now I'm going to switch gears and talk a little bit about how a clinical trial itself is conducted. And so there might be some variations depending on the drug or the type of disease or the phase of the study. But for the most part, all clinical trials are set up and run pretty much the same way. And so the first thing to know is a little bit of terminology in terms of who's involved. So the first term is what is known as a sponsor. So essentially this is an entity that decides that they have a research question that they want to answer. And so they decide to do a study, and they are ultimately the ones who would oversee and pay for that study. And so the sponsor decides how the study is designed, how all of the study data will be analyzed, and they're also responsible for making sure that the study is conducted ethically and appropriately. And so within the context of drug development, most of the times when we hear the word sponsor, it refers to a pharmaceutical company. They're just the most common type of sponsor out there. And then there are people who work at different hospitals or clinics where these clinical trials are actually conducted. And so collectively, we refer to these people as site staff. So usually there's, one doctor, one MD, who will serve as what's called the primary investigator. So that person is accountable for making sure the study is run correctly at that particular hospital or location. But essentially, all of the sites, they will follow the instructions that are given by the sponsor, and they're the ones who are actually seeing and treating patients within a clinical trial. And then obviously patients are, the people who have a particular condition, who sign up to receive an experimental treatment in a clinical trial. And so, they are required to come in at certain points to receive the experimental treatment and allow their data to be collected by the site and the sponsor. So the next thing to know is essentially in terms of getting started, the first thing that happens is that the sponsor will write a study protocol. So this is essentially a very, very detailed document that lists out every single little thing that needs to happen within a clinical trial. So it covers everything from, who's eligible to join a study. So for example, in a cancer study, the eligibility criteria might specify, it has to be adult patients, they have this type of cancer, it has progressed this far, they've received this many prior treatments, their kidney function is working this well, their liver is working this well. So they want to make sure that they're treating or bringing in patients into the study that can benefit potentially from the treatment. The protocol will also specify all of the data that needs to be collected and at what point in the study it needs to be collected. So here, this table, this is something that you would see in almost every study protocol. It's called either like a table of events or a schedule of assessments. But essentially, all of the rows in this table are the things that need to happen with the patient. So you can see things like getting blood pressure or blood work done or having a physical exam done. And then the columns are always when those things need to happen. So these are known as study visits. So here, for example, in this fake study, a patient would have to have their blood pressure taken at the first screening visit when they're being considered for the study on the 14th day of the study, and then at the very end of the study on the 49th day. So these are essentially the set of instructions that need to be followed for the trial. So after the sponsor writes the study protocol, assuming that this is a global study, which a lot of studies are, they take place in countries all over the world. So the first step would be for the sponsor to decide which country should be in the study. So part of this depends on the disease. Like for example, if a certain disease is more prevalent in certain races or ethnicities, then you would obviously want to target those areas of the world that have those patients. So once the list of countries is decided, then the sponsor would decide which sites. So again, these are hospitals or clinics. So which sites within each country they would like to have in the study. So for example, if I decide as a sponsor, I want the United States, I might decide I want Duke, I want UNC, I want Harvard. So that's kind of the process there. And the site selection process is based a little bit on whether the site can even run the study. So for example, if there are very specific tests that the study requires, not every hospital might have access to that test. So there are some operational considerations in picking which hospitals would participate in a particular study. But once the sponsor has all the countries and the sites, then they would send all of the study supplies and materials that the sites would need over to those sites, and then train all the sites on the study protocol so that all the sites are running the study the same way. So once the study is up and running at the sites, so each site is essentially always on the lookout for patients who might be good candidates to join that particular study. So once they find eligible patients, they would enroll those patients into the study, and then they would be collecting data from the patients based on that schedule of events I was talking about earlier. So they would collect specific data at specific time points. And this, again, is all defined in the protocol. So once the site has all of that data, the next step that they have to do is share this with the sponsor. So for every single visit and every single test and every single assessment, the site would be entering all of that information into a form that gets sent to the sponsor. So these are often called case report forms. This is an example of what a case report form might look like. And nowadays, case report forms are usually, they usually sit within an electronic database, so it's easier for sites to enter that data. So, as the study is running, all of this data is being collected by the sites are entering all of that data into the case report forms. So the sponsor is now receiving lots and lots of patient data. So now, once the study is over, the sponsor has all of this patient data from all of the sites, and now they have to analyze all of it. So what the sponsor will do is, they, in the protocol, they had specified what their analysis plan would be. So essentially, at this point, they just carry out that exact analysis. So there's different ways the data can be output. So on the left here is an example of a table. So literally, usually this just involves raw numbers of different areas of interest. So, you could have how many patients have to have their dose reduced, how many patients dropped out of the study. So there's a whole bunch of tables like this that would get generated. There's also a lot of graphs that get generated. So this is to provide a more visual way of understanding the data. So the graph on the right is a very common type of graph that we see in cancer studies, where essentially the faster the line drops to the x-axis, the faster patients are dying on that treatment. So here you can see there's two treatments, a pink and a blue one, but the blue one is dropping off not as quickly. So we can visually see that patients who are receiving the blue treatment are not dying as fast. So this is just an example, again, of a visual in terms of what would come out of the data analysis. So that's kind of, a lot about how trials are conducted. But, we do want to talk a little bit today about technology and, what place that technology might have within clinical trials. So, if you remember, I said earlier, it takes about 10 to 15 years to get one drug approved. So, in this day and age, I would like to think that we could do better than that. And this is because technology can make it a lot faster and easier to collect, share, and analyze data. So that helps save a lot of time and money. And this is important because there are still lots of diseases out there that don't have good treatments. So patients with those diseases are, desperately waiting for new and better treatments to come along. Also, technology is important because it can be really, really hard for patients to participate in clinical trials because traditionally patients have to go to their study site, so that hospital or clinic, for the site staff to collect data and information from them. And so for a lot of patients, this can be really challenging if, for example, their study site is far away and they don't have a convenient way of getting there. If they have to take a lot of time off from school or work to go for all of these visits, or if the visits are just very long and involve lots of tests and poking and prodding each time. And on top of all of that, they may not just be feeling well because of the condition that they have. So, this is important to consider because if too many patients drop out of a clinical trial, it can actually cause the trial to fail because not enough data is collected to test and verify the study hypothesis. So we want to see, hopefully that technology can increase convenience and reduce burden for patients moving forward. And finally, technology can also help us reach a lot of patients that might normally be overlooked for clinical trials. So, for example, a lot of times sponsors like to use sites that are these very large academic hospitals in big cities because, the doctors there are used to running clinical trials and there are lots of potential patients in that area. But this means that we would never have the opportunity for patients who are living in perhaps more rural areas to participate in a clinical trial. So we might not have a truly representative patient population in a study. And also, on the flip side, patients who might actually benefit from being in a clinical trial, they might never have that opportunity if they don't live close enough to a study site. So using technology is potentially a way to increase patient diversity, which is a really good thing for both the sponsors and the patients themselves. So there are tons of different types of technologies that are being developed and used within clinical trials. This is just a small flavor of some of the different types that are available. So for one, recruiting patients is really, really important for the sponsors and the sites because, drug development, again, takes a long time. We don't want to recruit patients so slowly that it's taking 10 years just to get enough patients into a study. So nowadays, a lot of sponsors are now doing things like creating websites to advertise their studies, and providing a lot of clear, detailed information to patients who might be interested in joining a study. So, these patients can go and browse these websites at their own convenience and consider whether or not they might want to participate. Also, a lot of sponsors are now using social media to advertise their studies and reach out to potential patients as well. And then in terms of logistics, so there is a lot of tracking and documenting that needs to happen within a clinical trial. So from everything from, shipping the drug itself and shipping all the study supplies, shipping any blood samples that need to go be tested. So there's now a lot of technology that helps manage and track all of those logistical kind of things. And then this is a huge area. So in terms of collecting patient level data, so I'll talk about this a little bit more in the next few slides. But we can use technology now to collect just unique types of patient data and also allow greater frequency of that type of data collection. And then finally, in terms of data analysis, there are people out there now who can create very advanced machine learning or statistical programming to take huge amounts of raw data and apply those algorithms to understand trends and essentially make sense of the data. So I wanted to mention a little bit about in terms of Datacubed Health and what we do. So this is where Pooja, Ashley, and I currently work. And so we are a mobile health company. We develop an app that patients who participate in a particular clinical trial can download. And so this is part of their study experience. And so our approach is that we use behavioral science to make clinical trial participation just a lot easier, and try to also make it fun, because a lot of times these studies are not necessarily fun to participate in. So we incorporate things like gamification. So this middle screen here shows a fun map where as you progress on your map, you're completing study assessments, and you're essentially going on a patient journey as you are going through the study. So it's just a different way of contextualizing the study for patients. We also want patients to associate their identity with the study. We find that this helps in terms of encouraging patients to continue participating in the study. So we offer things like being able to create your own avatar with accessories. And we also just try to create a very positive environment for these patients in our app. So there's a lot of encouragement and rewards. So for example, the screen on the right, every time they complete an assessment, they earn gems, which they can use to do things like upgrade their avatar. And so I did also want to mention a few unique pieces of technology that we use on our platform within trials. So one of them is called geofencing. And so what happens in geofencing is that in a given study, we would get a list of particular hospitals in a given area. And so we would take the latitude and longitude of those hospitals and plug those into our system. So anytime a patient who has our app on their phone enters one of these geofenced locations, we would capture that event. And then we could do things like send a chat to the patient to ask, hey, we saw you just entered, this hospital. Can you please confirm, are you here because you're not feeling well? So, this provides a whole new level of insight for the site staff and the sponsor, because, usually they don't really know what's going on with the patients outside of the clinic visits. And then another cool piece of technology that we use is telemedicine. So, this is especially relevant in this current time of COVID-19, but, telemedicine is really important in being able to provide patients with the convenience of being able to do study visits and study assessments at home, and still be able to connect with their doctors. So, this is something that is still relatively new, but it's something that we think will be, increasingly important as time goes on. So here I'm gonna stop sharing and give the screen over to Pooja on a demo.
Thank you for attending today's webinar hosted by Young Women in Bio-RTP. We hope you have gained an understanding of how therapies are approved for widespread use. An understanding of clinical trials is particularly important today when considering what steps must be taken for COVID-19 vaccines to gain approval and widespread use. We apologize for not being able to record the demonstration of the mobile app that Datacubed Health has developed. This mobile app includes proprietary information. Additionally, the recorded version of the webinar does not include any of the live question and answer due to some of the questions asking about proprietary information. In conclusion, we would like to thank our two speakers from Datacubed Health for their time. Thank you to Jennifer and Pooja for sharing their knowledge. We would also like to thank Datacubed Health for allowing us to speak briefly about their technology. Throughout the year, Young Women in Bio hosts many events for students to learn about different areas of STEM. Typically, these events are held in person. We would like to thank the sponsors listed on this slide that help contribute their time and resources to Young Women in Bio events throughout the country. As we continue to social distance, Young Women in Bio will continue to offer virtual events for girls throughout the country and in Canada. More information on these virtual events can be found on our Young Women in Bio online website listed here on the slide. We also have developed a Young Women in Bio online YouTube page that contains recorded versions of each webinar, as well as short videos that discuss particular STEM topics or careers. Volunteers throughout the country have developed a web-based series focused on COVID-19. These videos can be found on the Young Women in Bio online YouTube page. These are a great resource to learn about the biology and epidemiology of COVID-19, as well as what steps you can take to mitigate your exposure to COVID-19. If you are ever curious to learn more about science, please feel free to explore our Young Women in Bio online YouTube page. The Young Women in Bio online YouTube page will continue to be updated with new videos. This webinar was hosted by Young Women in YWIB-RTP. Events specific to Young Women in YWIB-RTP can be found on our Facebook page, which is listed here on the slide. We frequently update this page with our events. Please be on the lookout for new webinars in the future from Young Women in YWIB-RTP. Thank you again for joining us for this webinar. We hope everyone continues to stay safe. Remember, we are all in this together.